Okay, good morning, good afternoon, or good night. I am going to go through Lab 7, the genetics lab, with you. It's kind of difficult, I understand. Again, hopefully, you got to find some time to concentrate on those videos and questions and the reading, look up the vocabulary and such, but I'll do my best here, and I think I'll be able to give you a little lecture. You'll want to have some paper for notes. You'll also want to have a paper uh, to write a data table and to answer the questions for Genetics Lab 7. And you might want to have a partner, although not necessary for the lab. Again, I'm not going to do the whole lab here with you. I'm just going to introduce the lab, and then I'll do a little piece of it just to show you what you can do to do it. So here is our class, DC Biology. I go to classwork, and don't forget you have the lab manual. So you have to click on lab manual and open that up. Now, in reality, you don't have to open it up there. I want to remind, I clipped it right here. So, oops, apologize. Day 2 Genetics Lab. So here it is, Lab 7, click and open, and there it is. Go straight to it. Otherwise, if you want to open up the lab manual, you can. It's page 63. Just scroll down until you get there. So there's the lab, okay? Do a slow reading. Try to understand in light of meiosis, the laws of segregation. You'll see those here shortly. I'm going to talk about alleles and genes. I'm going to draw a picture to help make sense of this information. I'll reference these phenotype, genotype, and the homozygous and heterozygous condition. And then we'll stop on simple dominance. So I'm basically lecturing over this first page. And then we'll scroll down and we will do the simple dominance lab together, at least for a little while, because you'll notice in your worksheet, you're going to be flipping these coins 64 times. So I'm going to leave that to you. I'm just going to show you how you can take tally and tabs of this information. So this is one thing you want to have open is the biology laboratory. It might be good to come on over here and open up a Google Doc. All right. So I'm going to open up a Google Doc. Again, most of this you can take on your own notes. After you get a Google Doc, click on this. Now, if you have a phone, you'll need the app, the Google Doc app, which is not hard to download. It's very quick. Now, manipulating and typing in it might be a little more difficult. But anyway, no particular format. And you're going to just kind of fill in according to this here. So if you want to write this in a box, you can. You don't have to be a box. You could just type out heads equals TT equals tall, and then put the number, the actual number after you tallied it. That's what this total number is. So here you have like hash marks, right? Like, let me get my drawing tool out here. I believe I have. So 1, 2, 3. The next one was here, and then here, and then here, and then here. Actually, you'll probably get mostly here. You get the idea. You're going to have tabs, and then in the end, you're going to count it up 5, 6, 7. You had 8 total here. 1, 2, 3, 4 here, and 2 there. All right, so 4, 8, and 2. All right, so you'll just type out this information and this information. You don't need to put this into the Google Doc. What is the phenotypic ratio of tall to short plants? Well, tall and tall, these are the phenotypes. This is a phenotype. This is the genotype. And so I see I'm going to have 8, 9, 10, 11. I'm going to have 12 tall ones. And I put dot, dot. That's the way we do a ratio. And I only have 2 short ones. Well, we see that 12 is divisible by 2, so you could really say 6 to 1. And that's what you would put there. Phenotypic ratio, you just type out 6 to 1 if this is your data, okay, which I doubt it will be. Whoops. Okay, so we're gonna do all this. All right, let me go ahead and clear my drawings, and let's back up now to the lab itself. Okay, I said to the lab itself, but really I wanted to do that lecture first. So, all right, so this is once again lab 7 lecture. All right, that's a C, not an E. Okay. All right, so let's just kind of back up a little bit, and we know what an animal cell looks like. Kind of irregular, it has a nucleus, and in the nucleus we have all this DNA, correct? Now DNA is a double helix, but that's what it looks like really up close and such. And generally what we see with DNA when we look is like all this long lines, okay, beads on a string. But you might recall that in meiosis and mitosis, this long strand of DNA here becomes coiled up real tightly into chromosomes, right? We call this supercoiled. There is an end to it, and usually somewhere in the middle it's really dense, and this was called the centromere. Can I hear an amen? Do we remember this? The centromere. So this is a chromosome. And we generally represent the chromosome simply as being something like this, where we have the centromere, again, right there, and this is the chromosome. Now, if you remember in the cell cycle, okay, and remember this is a recorded video, so if you need to pause at any time, you can always pause and resume me. So these are the phases of mitosis, and we had the prophase, metaphase, anaphase, and telophase. Here's G1, S, and G2 phases. So this is what the DNA or chromosomes would look like right here on this side of the S phase. But once we go through the S phase, the chromosome will have duplicated itself in preparation for division by mitosis. So that once it goes through mitosis, we'll have 2 new cells, each with one of these copies right here. And one will be in here, and one will be in here, right? So that's the purpose of the S phase. Duplicate the DNA, make a copy of it. These are now sister chromatids. These are sister chromatids. The whole thing is also called a chromosome, a duplicated chromosome. Okay, so this is the background you need coming into today. What we want to talk about today are going to be the genes, okay? G-E-N-E-S, G-E-N-E-S. Genes reside all down here. Remember, a chromosome is what? This long strand of DNA here. It's just been coiled up really tightly. So maybe from right here to right here codes for a trait. Well, that might be a long stretch of DNA, but when it's tightly coiled, it only takes up that much space. And so we generally let lines, if it's a big gene, it might be a fat line, might be a thin line, barely, so those marks across the chromosome represent the genes. And what is a gene? It's a segment of DNA that codes for a trait. Oh, shoot. All right. So now I'm going to do some of that again. Here's our cell once again. A regular cell with the nucleus. Okay. So we understand when I do this now, and we're talking about a chromosome and the centromere, that we have genes in various places on that chromosome. Okay, so each of those lines represent genes. So I'm just going to draw an arrow here. I'm going to say that represents a gene. Okay, so if this is a haploid at this point, we only have one set, correct? So if we had 2 sets of those, we would call this, and again, it has to be identical. Is it 3 lines. This would be the diploid, right? Here we had one copy of a gene, or I'm sorry, one copy of a chromosome, and here we had 2 copies of the chromosome. So this would be a diploid, right? You'll notice that these chromosomes are about the same size and they have the same banding pattern of genes. Not every chromosome does. But let's think back to how we got this diploid set of chromosomes in a single cell. Okay, if this is diploid, let's think of this as being mother's egg. If it undergoes meiosis, what happens here? Well, you're going to get 2, right? And ultimately, you're going to have 1 chromosome come into each of these, and you're going to get 2 haploid from that 1. Now, this is not an exact replication. Of course, there would be 4, right? But I just want you to understand the point that we're reducing the number of chromosomes. And it's not going to be the best drawings either. And so if this were the male, right, diploid, and he also had 2 sets, 23 pair of chromosomes. Let's call this chromosome number 13 for fun, okay? We have 23 sets. We number them 1 through 23, and we have 2 sets of 23 to make 46 chromosomes is a diploid, all right? Diploid in humans is 46 chromosomes. This will undergo meiosis as well in the gametes, the sex cells. And now we have 1 chromosome here as well. These chromosomes code for the same trait. That is, all the genes on this chromosome code for the same trait. And once again, we have haploid and haploid. I'll reiterate that in a second. So in fertilization, we make a new cell that will then have 2 identical chromosomes. And now this is diploid, and that is you, right? That's you. So this is where we're going to pick up from. Let me back up real quick. In a cell, you have 23 chromosomes in your sex cells. Each chromosome has hundreds to thousands of genes located on them because that chromosome is really just DNA. All right. Now, you'll notice that the way you got to be diploid, say down here, was this process of fertilization. So if these are sex cells, they underwent S phase in the cell cycle, and so there's 2 copies of each chromosome now. So he and she bring in a diploid set, and their sex cells will undergo meiosis. The law of segregation says these 2 chromosomes will separate from each other, and they did. And they did so independently. Likewise in him, the sperm production, and so now you have sperm, right? These are sperm and egg in her. They fertilized and made a brand new diploid zygote. So what we're interested in now is we're getting away from this, and we're going to look at how inheritance occurs and how these genes are separated, okay? So I'm going to pause the here real quick. All right, sorry about that. So now let's pick up. You can just continue in your notes if you're taking notes, but let's remember what we got here. All right, we had the zygote. Right, that was diploid because it had 2 chromosomes, 1 from each parent. And we're just looking at 1 homologous pair. Homo means same, homologous. This is 1 homologous pair of chromosomes. Homologous pair. That they're homologous means they code for the same trait. So if there's a gene here, there'll be a gene here. If there's a gene up here, there'll be a gene up here. Wherever there is a gene. So we got to make those 2 chromosomes look alike. So let's talk about now if we have, remember we had 1 chromosome that became after S phase 2 chromosomes, right? So if this one had chromosomes that look, a banding pattern of genes that look like this, then of course, the copy is going to also look like this, right? So you can see that. And then once they separated in meiosis, each of these sister chromatids were separated from each other to form the gametes. And one of these got to fertilize or was fertilized by another. Okay, so this is where we're at. We're looking in that cell that was here, that was that had them. So what I'm saying is, you could call both of these, all of these, genes, because they are generally genes, but they don't necessarily code in the same way. So a gene, we'll just say codes, whoops, come on now, codes for a trait. It's for a trait. Okay, genes code for a trait, but not all traits are the same. Okay, so when we say not all genes are the same, what we mean is there's different versions of a gene, correct? So let's see what I'm talking about here. There's a term we call allele. Alleles are alternate forms of a gene. Okay, they're alternate versions of a gene. Allele is an alternate version of a gene. Sometimes there's only two versions. Sometimes there's many. What is it, for instance? Well, in the video of heredity, you saw that you could have wet earwax or you could have thick, waxy earwax. That's an either or. Widow's peak on your hairline or no widow's peak. That's an either or. There's only two versions of that gene. Eye color, for example, would not be, because you can have blue, green, hazel, brown, gray, and some variations of speckled in between, right? So it's still coding for eye color. All right, it's still coding for eye color. Whoops, I want this. I'm going to make some room here. If you understand that you have two versions of every gene, then we're good to go. And those two versions of the gene come, one from your mom and one from your dad, and they're inside you. So this allele might say green eye color, whereas this allele might say brown eye color. Now they're both coding for what? Eye color. So the gene or the allele is eye color. Okay, the two versions of the genes, that is alleles, that's two L's, excuse the bad handwriting here. All right, so the genes, eye color. So the two versions of the alleles are what comprise your genotype. That is a type of genes. Okay, your genotype. What you actually look like, okay, is called the phenotype. That's your physical expression. That's a T. Phenotype. Okay. So I have a phenotype is a physical appearance, the actual traits you have, and it's based on what? A genotype. Well, what if you're in this case where both of your alleles are different? You have an allele that is a version of a gene that's brown, and an allele, a version of a gene that's green. Well, what do you get? Some mix, possibly, in this case. But before I go on and tell you, I just want you to see that you have, even if there's multiple alleles in the human population, as we just said, blue eyes, green eyes, brown eyes, hazel eyes, you know, in the human population, there's many alleles, but every human being, individual, has only two. The one allele they inherited from their mom and the one allele they inherited from their father, pop. So those two versions that you received are what's called your genotype. The two versions, the genotype are the two versions of the gene. Phenotype is the physical expression of that. So I'm going to clear this drawing and rewrite again. You know, I don't think I want to draw these chromosomes anymore. I'm only going to be interested in this gene, okay? So again, we have an allele here. Now, how do we represent it? We do know, just by observation, that there are more brown-eyed people than green-eyed. So it's most likely that the allele between those two, the allele for brown happens more often. So it's probably dominant. Whenever that phenotype, when that physical appearance occurs more often, we call it dominant, and we give it the initial. We represent, and we'll usually do a capital B. The allele for green, all right, then we cannot change the letter because it's the same gene. Remember, it's just two alleles, two versions of the same gene. So we still got to use the letter B, but it is recessive, so we use the lowercase b. So the way this is represented, I mean, we could also just do the two chromosomes as, you know, in a cell as just two lines, and there's the centromeres, and here's the lines, and we say capital B and little b. And you understand by looking at this picture right here that you're looking at a cell, and for this particular trait of interest, the particular trait of interest, eye color, you can see you have two different alleles. You have an allele for brown and you have an allele for green. Whenever this happens, an individual has two different alleles, we call that heterozygous. Heterozygous. Okay, and in the heterozygous genotype, the dominant will always express itself. And so even though the genotype is heterozygous, and this is how you represent the genotype, even though the genotype is heterozygous, the phenotype cannot be both. The phenotype has to be one or the other, and if it's always the dominant, then it's going to look like the dominant, and the dominant in this case is brown. And so whenever we ask for a genotype, we're looking for letters, a combination of letters. So if it's a genotype, we want to know the type of genes, and those are represented by letters on the chromosome, okay? And we want to know a phenotype. Think of that, pH is physical phenotype. Now we're asking for the actual color, and the actual color would be brown, okay? So a phenotype, so we have alleles. Alleles are versions of a gene. Genes code for traits, okay? Genotype, as you see in the lab, is the combination of alleles, the combo of alleles that you have, okay? And a phenotype is the visible expression, okay? In other words, for the most part, what you actually see. I can see you have a particular color of eyes. I cannot see what your genes are. I can't see your genes, but I do see your phenotype. And so based on your phenotype, I can possibly figure out what your genotype is, but I couldn't know with certainty. Okay? And now then, lastly, if your genotype, so these are possible genotypes. I'm going to write here. And there's an easier way to refer to this. If you have two of the same genes or two of the same alleles, then you are what you call homozygous. And in this case, it's homozygous. You have to say, well, two of the same what? Well, there'd be two big ones. So it's homozygous dominant. If it is little b, it is also homozygous, because homozygous, that means in the zygote, you have the same traits. But now, if they're the same, you have to tell us, are they the same and they're recessive, or are they same and dominant? So this is what you call a homozygous recessive genotype. This is what you call a homozygous dominant genotype. And what if you have one of each? Well, then you are heterozygous, okay? Some of your traits you inherit from your parents are the same. If both your parents have black hair, or both have brown hair, or both have, you know, same eye color, then you likely may be homozygous. But sometimes you might have a parent with brown and a parent with black, or blonde and black, or blonde and brown. Well, it's most likely you're going to be some mix in between, and that's what you call heterozygous. And with that, you have two different versions of the allele. One you got from your mom, one from you got your dad. That should cover all the terms in the lecture. I hope that helps understand, or at least on that front page. My lecturing skills should get better as we go through. But until then, you have this to deal with. Okay, I'm going to make a separate recording to do the actual lab.